This is the last lecture that I'm going to post the PowerPoint presentations for, because the last 2 lectures are optional. If you don't want to go to them, and I understand it has been a long semester, that's fine. If you want to go to them, I want you to sit there and just listen. But that information will not be on an exam. This is the last information you will be responsible for in an exam. So today we're going to talk about the proteases. And if by chance you get interested in the material and want more in depth, there is chapter 10. It's on Canvas. We wrote this chapter, peptidases. There's some really good stuff in here. And the first sentence starts, of all known enzymes, peptidases are high up on the biochemical and medical priority list. They really are, and they're important. So what we're going to do is start off with an introduction today, and then we're going to turn our attention to the serine peptidases, then to the cysteine peptidases, and the metallopeptidases. And then the last category, asparto and glutamopeptidases, we will save for the last lecture. So today you're responsible just for the serine peptidases, the cysteine peptidases, and the metallopeptidases. Now, the IUPAC wants us to refer to them as peptidases, but I have a tendency proteases. It's a little bit simpler. But peptidases are proteases, and there scarcely is a form of life that does not have at least one peptidase associated with it, and that includes viruses. So they're medically relevant, very medically relevant. They're also referred to in the literature as proteinases or proteases, and I like the term protease. Now, they've been studied for more than 150 years. 150 years. They are important in the biotechnology field. They're used, for example, in the production of animal feed, the production of textiles, the leather, the food industries. Meat tenderizer. If you use meat tenderizer, you're using a protease. They're also used to clean off stains off clothing. Now, prokaryotes use peptidases mainly, or proteases, that's why I like the term protease, use proteases mainly for the degradation and recycling of proteins. But that's not true for eukaryotes. So, for example, most of the 500 or so mammalian peptidases are involved in a very limited and specific proteolytic event, and we'll talk about those. So let's have some generalization about the peptidases. One thing you should know is these enzymes are synthesized as inactive precursors, zymogens. And why is that? Because they chew up proteins, and you don't want them loose until it's ready for them to start chewing up proteins. So that's important. They all come as zymogens. And the zymogen conversion to the active form is interesting. It can be just a change in pH. It can be autocatalytic, depending on whether an adapter protein is binding to it. So there's all different methods of how these proteases are activated. The size of the activation segments range from dipeptide units independently to folding domains that are 100 residues. So it's really remarkable, some of them. A common form of activation segment is an N-terminal extension of the mature enzyme or prosegment that basically blocks the active site, and when that gets cleaved off, the active site opens up, and then the enzyme can function. So as I said, how you activate them is stunning, and it's different, as it could be just a drop in pH, or it could be interacting with another physiologically important factor, so it depends. But what all proteases do, what they all do, is they catalyze transfer to water. That's what they do. It's group transfer to water. Now, when you think about it, the concentration of water is 55.5 molar. There's a lot of water around. But this breaking of the peptide bond is actually very, very slow. It's a strong bond. And without these enzymes, the uncatalyzed hydrolysis of a peptide linkage, the half-life at neutral pH at 25 degrees Celsius is 300 to 600 years. So that's why these things are so important. You can't digest proteins. The 4 major classes of peptidases. The first are the serine peptidases, and those are the ones, honestly, we know the most about. Those were the ones that were first studied, and those are the ones you learn about in introductory biochemistry. They're inactivated by diisopropyl fluorophosphate, and they're also inactivated by chemical warfare agents like soman and sarin. We've talked about soman and sarin before in this course. The cysteine peptidases are inactivated by iodoacetamide or iodoacetate, and we talked about that in the beginning of the class when we were talking about amino acids and their personalities and what kind of reagents react with their side chains, and that's the cysteine peptidases. Now the metallopeptidases, obviously they use a metal, and so you can inactivate them by, for example, EDTA, which chelates the metal. And then the alkaline or the aspartoglutamyl peptidases, they are not active at neutral pH or slightly basic pH. And so you can inactivate them by changing the pH. Those are the ones we will talk about on the last day of class, if you attend. Now, there are so many peptidases out there, or proteases, and how do you describe all of them? In the beginning, when they were first studying chymotrypsin or trypsin, they didn't know very much, but now we know there's hundreds of these peptidases out there. And how do you see the forest through the trees? How do you do that? So there's lots of classification schemes. I'm not big on classification schemes, but it's important that you understand they are out there. So there's a whole book, Handbook of Proteolytic Enzymes by Rawlings and Barrett, and it's called MEROPS, and what they do is they put the different proteases out there in terms of family, clans, and catalytic types. So I'll show an example of that in a few minutes. And if you're interested in that, or you read a paper and they're talking about it belongs to this clan or whatever, you can take a look at this book. But what this doesn't do is tell you anything really about what the peptidase is doing. So I like this particular classification scheme. You don't have to memorize all these, but you know, some of them are pretty easy to remember. So if you hear the term endopeptidase, well, it's hydrolyzing internal bonds away from the N or C terminus of the protein. Endopeptidase, that's pretty easy to remember. Omega peptidase, what's that one? Probably haven't heard about that in 507 or 501. It's distinct from an exopeptidase in that it does not require a free N or C terminus. So you can have a blocked N terminus or a blocked C terminus. And we've talked about those kind of modifications that occur. Exopeptidase makes a lot of sense. It's chewing away at one of the termini. So it requires a free N-terminal amino acid group, so you can't have a blocked N or C terminus. Aminopeptidase, what is that one? We're going to talk about an aminopeptidase. It liberates a single amino acid residue from an unblocked N terminus. A carboxypeptidase, what do you think? It's working at the C terminus. It liberates a single residue from an unblocked C terminus. Dipeptidopeptidase, it removes an N-terminal dipeptide from its substrate. Tripeptidopeptidase, it removes an N-terminal tripeptide from the substrate. And remember, the substrates are themselves peptides or proteins. That's what you got to keep in mind. Peptidyl dipeptidases removes a C-terminal dipeptide from its substrate. And dipeptidases hydrolyze a dipeptide. The other thing about these substrates is they're not little molecules. Right? They're peptide bonds, they're peptides that are coming in, so the active sites tend to be open, and that's going to be an important part when we get to another couple of slides. So what are the common characteristics of all these different types of peptidases? They all go through an intermediate or a transition state in which the normally trigonal carbonyl carbon, which is sp2 hybridized, that's why we talk about this on the first day of class, becomes tetrahedral. So it goes from sp2 to sp3, we go from trigonal to tetrahedral, flat to tetrahedral. The tetrahedral geometry occurs because of the temporary addition of a nucleophile. And the oxygen, when it goes into that tetrahedral intermediate, the oxygen is carrying a negative charge. That's the oxyanion. And what these proteins have is something to basically stabilize that oxyanion. It's called an oxyanion hole, and we'll talk about that. In the case of the serine thiopeptidase, it's the amino acid that's directly going after that carbonyl carbon. And then you end up with an acyl intermediate that has to be hydrolyzed off. Whereas the other two, the metallopeptidases or the aspartopeptidases, use water. And the reaction proceeds more directly in the peptidases, metallopeptidases, and the carboxylpeptidases, these aspartoglutamyl peptidases. Whereas an acyl intermediate occurs when you have the serine attacking or the cysteine attacking. Now, one of the things they don't teach you about in 501 or 508 is the Schechter-Berger nomenclature. And if you're working in a drug company and you're interested in basically targeting a protease, you have to understand this particular nomenclature. And this nomenclature was put back in 1967 by Schechter and Berger. Now, I would have done it differently, but they're the ones who put it out in the literature. So let me explain this slide, because this took me a while. Once I actually was a postdoc in Eugene, Oregon with Brian Matthews, and I worked on thermomycin, and I had to learn this nomenclature, because thermomycin is a protease. All right, so here, let me describe this. It's N-C alpha C double bond O, N-C alpha C double bond O, N-C alpha C double bond O. So we're coming along here. Here's our substrate. It's a pretty extended substrate. This is the bond that's going to get cleaved. That's called the scissile peptide bond. That's the one that gets cleaved. Now, anything to the left of the scissile peptide bond, it's called the P1, so that would be a side chain coming off. That's the P1 position, P2 position, P3, and P4. So that's to the left of the scissile bond. Anything to the right of the scissile bond is called the P1, P1 prime, P2 prime, P3 prime, and P4 prime. Now the P stands for peptide, or protein that's going to be hydrolyzed. Now I would have called it S for substrate, but that's not what they did. Now these S refer to the sites that the peptide comes into and binds into. So this is the S2 site on the protein, the S4 site here, S3 site, S1. Anything to the left of the scissile bond is S1 site, S2, S3, and S4. And what you have to visualize in 3 dimensions, that there's side chains in that S1 site that's going to allow specificity of the P1 side chain from the substrate coming in. Right? So the P stands for the substrate, and the S stands for the protein's binding pockets. So they did it reverse of what I would have done, but it's been in the literature since 1967. What you have to know is that some proteases have really, really good specificity where they will only cleave a peptide bond if, for example, that P1 is a hydrophobic residue or an aromatic residue. Some enzymes, they don't care, but some are specific. And why is the difference? Because think about the 3-dimensional structure. Think about those side chains there. How this pockets that are going to bind the substrate, how are they arranged, and what kind of residue? Can you have a big residue, a small residue? Do you have a charged residue in the P1 site? Schechter-Berger nomenclature, important. I'm going to reiterate again. If you're on the left-hand side, it's P1, P2, P3, left-hand side of the scissile bond. If you're on the right-hand side looking at it, it's P1 prime, P2 prime, P3 prime. So let's move on to the serine peptidases. They are by far the best understood. They were probably the easiest to start working with, and so we understand that they've been examined more than 50 years. And what they all have is this peculiar serine residue that is really reactive. Now, there's serine residues all over the place in proteins. What is it about this serine residue that makes it so reactive? That's what's fascinating. There are 3 distinct structural families of serine peptidases. They have very different structures. And yet, when you look at them, they have very similar catalytic mechanisms involving 3 residues. And it's suggested that this is probably one of the most striking examples of evolutionary conversion, where nature converged on this, what's called catalytic triad, a number of different times. So the overall structures are different, but the way these 3 residues, this catalytic triad, is lined up is stunningly similar in all 3 family types. So one family is represented by the bacterial peptidase called subtolysine, and we're going to talk about subtolysine. Another by enzymes that include chymotrypsin and trypsin that they always tell you about in introductory biochemistry. There are the kallikreins, for example. These are serine proteases, and you probably have heard of the prostate-specific antigen, or PSA test that they do. Well, they're actually looking for kallikrein 3, which is a serine protease. And then there's a final family represented by wheat germ serine carboxypeptidase 2. Now, the trypsin family, they're involved in digestion, blood clotting, for example, fertilization, complement activation, hormone production. They're very widespread. So let's again talk a little bit about the serine peptidases. As I said, this is the cis-aryl bond right here. Anything to the left of it has a P1, P2, P3, P4. Anything to the right is P1 prime, P2 prime, P3 prime, and P4 prime. Most have a striking preference for what's sitting in that P1 position. Not all, no. Most of them do. And trypsin, you learned about Pleiades bonds only at lysine and arginine residues, because if you look at the site where that residue fits in, where that P1 goes in, there are charged residues there. Chymotrypsin after a large hydrophobic residue. Some of the other peptidases have less distinct preferences. Now this is important because this is something they don't teach you, but we know that the peptidases can hydrolyze amide bonds, peptide bonds, but they can also hydrolyze ester bonds. And so if you're in a drug company and you're trying to design something to inhibit a peptidase, and you put an ester bond in there, guess what's going to happen? It will hydrolyze it. Now these are some of the inhibitors that inhibit serine peptidases. There's diisopropylfluorophosphate right here. We know about that one. And as I said, serine and soman as well. What do all the serine peptidases have in common? They share an oxyanion home. That's important. And they all contain this catalytic serine, histidine, aspartate. Serine, histidine, and aspartate. Histidine and aspartate. So what happens is the enzyme, and this is the serine, it's going to attack the carbonyl carbon here of the peptide bond. You have an acyl enzyme intermediate right here. It's tetrahedral right here. Sorry, it's tetrahedral. That collapses, and then you have the acyl enzyme intermediate. That has to get hydrolyzed off. So all these go the same way, and it's the serine that attacks. It's the serine. But remember, serine, if it's not hydrolyzed, it's not an alkoxide, it's not a good nucleophile. You got serines all over in proteins. So what is it about this one? What is it about this one? So the thing to remember, the serine's going to attack. You have a tetrahedral intermediate and a transition state. So if you're interested in drug design, you might think, well, if a transition state is tetrahedral, maybe I can design something that mimics that tetrahedral transition state. And then that collapses off, and you have this enzyme acyl intermediate that has to be hydrolyzed. Now, this is sublysin-Carlsberg. This represents one of these categories of serine peptidases. And by the way, this is an important detergent enzyme. So this is one of the enzymes they use in probably Tide. I'm not sure which ones actually use it, but this is the enzyme and it comes from a soil-dwelling bacterium. So it's a central parallel beta sheet surrounded by 4 helices, 2 on each side. So here is your sheet. Right here, you have 2 helices on this side, 2 helices on this side. You have some more shrubbery in this. This belongs to the family S8 clan SB. That's the Merops. And what that really doesn't help you, except this S pretty much tells you it's a serine protease. It has broad specificity. It's an endopeptidase, which means it's going to cleave bonds internal, away from the N and C termini. So that's what the structure of sublysin-Carlsberg looks like. And again, this is used in laundry detergents. You have an aspartate, a histidine, and a serine sitting there. That's called the catalytic triad. Now let's go ahead and look at bovine gamma chymotrypsin. Completely different structure. It has antiparallel beta barrel. It has a Greek key motif, no less, in each an antiparallel beta barrel type here. But the important point is this is family S1 clan PA, not that that matters. But look at this, it's a aspartate, a histidine, and a serine. There you go. It's an endopeptidase as well. And the preference is for a phenylalanine, a tyrosine, or a tryptophan in that P1 site. So when these structures were solved, especially chymotrypsin and trypsin, they came to call this called the catalytic triad. This arrangement is known as the catalytic triad or the charge relay system. Now the earlier literature suggested that this histidine pulls that proton off that serine to make it an alkoxide. And we know now from this class, if you have an alkoxide, you have a strong nucleophile. But what are the chances of that? The OH group, right, it has a pK of what, 15? And the histidine, what's the pK of histidine, 6? What are the chances of doing that? And the answer is no. But these were crystallographers solving the structures. They weren't enzymologists, they weren't chemists. And so they've had this thing in the literature how this aspartate pulls the proton off from the histidine pulls the protein off, or pulls the proton off here, and you have an alkoxide. Well, we know chemically that doesn't work. But what we now know, it's in conjunction of these three together that allows this to happen. Now, what's stunning is that if you look at the serine proteases, they have these catalytic triad, and if you line them up, they align perfectly, even though the three-dimensional structures are completely different. So, serine peptidase, three distinct structural families. There's the chymotrypsin family right here. There's the subtle lysine family right here, and then there's the wheat germ carboxypeptidase family here. But look at this, the aspartate, histidine, serine, aspartate, histidine, serine, aspartate, histidine, and serine. And they don't even occur along the amino acid sequence in the same order. So chymotrypsin, it's 102, 57, 195, but in subtle lysine, it's 32, 64, 221. So if you line these up, they line up. What it's telling you, nature converged upon this catalytic triad a number of different times. So what's the proposed mechanism for these serine peptidases? So here comes your substrate coming in here. This is the cis-aryl peptide bond that's going to be broken here. This is the carbonyl-carbon that's going to attack. Now you have to remember, this is all drawn as a catalytic reaction, but you've got to envision these peptidases and how they actually fit into the active site. How does the substrate get in there? Because they're long extended substrates. Right, so here you have this serine here. It's activated by this histidine and aspartate, so you're kind of pulling along the protons on here. But this then attacks the carbonyl-carbon here. This does not attack as an alkoxide, because that just doesn't make any sense. But it's this kind of pulling the protons along that you end up with a tetrahedral intermediate here. All right, and here's your oxyanion. So now you want this to collapse. But this group, this NH group, is going nowhere unless you put a proton on here to make it a better leaving group. And so what happens is the histidine that grabbed that proton off the serine is now going to put it onto here, and what happens? It collapses. And so now you've broken the system on a peptide bond, except you have an acyl intermediate right here. So then what comes along is a water molecule comes in here, and the aspartate's pulling the proton off the histidine. The histidine is going to help to pull the proton off the water molecule, but it doesn't completely pull it off. You can't have a hydroxide going in. It's the concerted effort of these that allow it then to attack the carbonyl-carbon here. This is now, remember, attached to the enzyme. You end up with another tetrahedral intermediate. And how are you going to get that tetrahedral intermediate to collapse? Well, this isn't going to leave. This serine group that's now holding on to the substrate isn't going to leave unless you protonate it. So this bond has to get protonated here. And now you've set up your catalytic triad and you've broken the peptide bond. And it's fascinating. When you think an uncatalyzed cleavage of a peptide is 300 to 600 years, it's remarkable how these enzymes do this. Now here's the oxyanion hole, and what happens is in the Michaelis complex, in comes the substrate. This is the cis-aryl peptide bond that's going to be broken here. This is the P1 site here. And what you see is there's an oxyanion hole where there's backbone amine group hydrogens pointing down here. And then when you go into the tetrahedral intermediate transition state, what you can see is these peptide amine groups pointing down help to stabilize that tetrahedral intermediate and then oxyanion. So you learned about chymotrypsin, you learned about trypsin in basic biochemistry classes, but let's talk about one that's medically relevant. And this is the neutrophil elastase, human neutrophil elastase. It's also called HNE. Now, this peptidase has a lot of activity. It's important. So it has intracellular activity. So it involved in the degradation of foreign proteins during phagocytosis of neutrophils. And what are neutrophils? They're types of white blood cells. So they take in something, and then this serine peptidase breaks it down, foreign objects. But they also have extracellular activity, so they can be secreted out. And their extracellular activities include degradation of elastin, immunoglobulins, clotting factors, all those kind of things. Now you think about it, this human neutrophil elastase, HNE, has to be inhibited because you can't have it chewing up stuff, right? So In the lungs, it's normally inhibited by this thing called a serpin. A serpin is serine protease inhibitor. Now the serpin is 394 amino acid residues in length, and it inhibits this human neutrophil elastase in the lungs, so it doesn't start chewing up the lungs. But what happens with smoking is it leads to a lower than normal levels of the serpin, and it's suggested that it actually contributes to emphysema, because you're taking the serpin level away, and that lets the human neutrophil elastase start chewing away. So it's been implicated in cystic fibrosis, it's been implicated in rheumatoid arthritis, it's been implicated in the development of COPD, which is chronic obstructive pulmonary disease. COPD is probably going to be the third leading cause of death in 2013. And so you can pretty much understand how this, or 2013, how this is a target for a drug design. Because if you can inhibit it, you might be able to stop the advancement of some of these diseases like emphysema. Here's an example of a compound that was put forth. Now, it never made it into clinical trials, but the point of showing this is, in fact, that you can see how they're designing drugs. They're looking like they're peptides, it's N-C-alpha-C double bond O, but then they put in groups that can't get hydrolyzed by the enzyme. And so there is a lot of work being done, and here's an example of the structure of this enzyme being solved in the presence of this compound right here. There's the aspartate, histidine, and serine. It is a competitive inhibitor. So the serine peptidases, we know a lot of information about the serine peptidases, and we're learning a lot more about the cysteine peptidases as well. They are widely distributed in nature. You have plant enzymes, papain. The South American cooks knew for many, many years before the tenderizer industry got involved that if you took papaya juice and put it on meat, it would tenderize the meat. They didn't know why, but they knew it would work. So papaya uses a meat tenderizer. Bromelain, it comes from pineapple. That's if you use Adolph's meat tenderizer, that's what you're using. So there's cysteine peptidase. Actinitin comes from kiwi fruit. There are 5 additional superfamilies. You don't have to know all these, but the important point is they all have in common a histidine-cysteine pair. A histidine-cysteine pair. And cysteine, you remind yourself when we learned about the personality of those amino acids, the cysteine can get hydrolyzed, or not hydrolyzed, can let off its proton and become a thiolate. So, histidine-cysteine combination. That's important. And this is a slide that came from the first lecture, or the second lecture, I think it was the second lecture when we were talking about amino acids. So, we know that cysteine, the side chain, if it's a thiolate, and it can be a thiolate if you've got a histidine sitting there pulling it off, because the pKa's of the histidine and the cysteine are much more similar than they are between a histidine and a serine. That's an important point. So it can attack iodoacetamide, or you can end up with malignamide right here, and you end up with a thioether in these cases. So you can inhibit these enzymes. You can inhibit cysteine peptidases by these types of compounds. Now, we could talk about something like actinitin from kiwi fruit, but I thought we would talk more about programmed cell death, or apoptosis, which is something you learn in cell biology or basic biology classes. But it all boils down to cysteine peptidases. So there's a lot of, when you see these classes and there's this circle going to this triangle to go this square, and they form this complex, and on and on and on, it still has to do with cysteine peptidases. Cysteine peptidases. So let's talk about programmed cell death, or apoptosis. The ability to commit suicide is a fundamental property of animal cells. Now that, when you're studying, you never start to think, well, wait a minute. Maybe because you see something die, you think, well, why would a cell want to commit suicide? Well, if you think about it, 10 to 11 to 10 to the 12 cells are produced every day in a human adult, healthy human adult. Can you imagine what would happen if you didn't kill off the same number of cells? But it was proposed back in 1972 that there was a programmed cell death, that cells, there was some genetic thing or something that said, it's time for you to go, it's time. But it took so many years for people to really begin to believe that that was important. So apoptotic cells, what they do is they shrink, and then they're rapidly eaten by other cells, And the point is they don't let out their contents, which could really cause problems about neighboring cells. This idea that animal cells have a built-in death program was crucial insight back in 1972, but it took 20 years. It took more than 20 years for people to accept it. And as I said, as scientists, we want to understand how normal cells function. We never gave it a thought. It's like, well, normal cells function finally by saying it's time to go. So, apoptosis versus necrosis. Right? Apoptosis is an orderly way of saying it's time for this cell to go. There was some cell damage or it's just time. And it's an orderly way of getting rid of a cell without destroying other tissues around it, without destroying other cells around it. Now, necrosis is different. Right? That happens with infections, for example, diseases, all sorts of issues. So necrosis is something you don't want. That causes cell death in big times, in big sheets of cells, where in apoptosis, you're killing a cell at a time. And it's a very complex mechanism, and it has to be when you think about it. You don't want cells committing suicide unless it's time to go, unless there's a signal that says it's time to go. It's a very complicated and tightly regulated system. So it involves death signals inducers, it involves receptors and adapters and regulators and other regulators, but what you have to understand is it ultimately comes down to cysteine peptidases. Cysteine peptidases. Other facts, right? Programmed cell death depends on protein cleavage. That's clear. Now, it all depends on these things called caspases. So apoptosis, there's a lot of cell biology in it, but it all comes down to caspases. And where'd that name come? It's a cysteine peptidases, and the P1 position that it likes is an aspartate. So it's caspases. C stands for the cysteine peptidase, and aspase refers to the cleavage of an aspartate residue in that P1 position, the Schechter-Bergen nomenclature. There are two major pathways for apoptosis. There's the internal and the external. Now the internal, it's called the intrinsic pathway. And basically, there's something happens. Maybe there's radiation damage or something. There's something internal to the cell that says it's time to go. And so it's the mitochondrial pathway. And when this happens, the mitochondria basically release cytochrome C into the cytoplasm, and that triggers this intrinsic pathway. The second pathway involves the ligation of death receptors at the cell surface. And that's sending a message external to say it's time to go. And that starts this whole extrinsic pathway. Where does apoptosis come from? It comes from the Greek name, which refers to the shedding of petals, or from flowers or leaves in the autumn, when we've seen a lot of apoptosis this fall. Intrinsic versus extrinsic pathway, and of course you don't have to memorize, but to me, it's fascinating how tightly coupled things are, and they have to be. Sometimes when you listen to these lectures, you walk away and going, how does this all work? How does it work? So intrinsic pathway, there's radiation, there's something to stimuli that says, it's time to go. And so the mitochondria lets go of cytochrome C, that gets into the cell here, interior part, you start forming these complexes, and ultimately what you're doing is activating cysteine peptidases that activate other cysteine peptidases and then start breaking apart the cell. That's what you're doing. In the extrinsic pathway, there are some death ligands that come along. They bind to a receptor. There are adapters there in the cytoplasm, and then they come along and they form these complexes that then activate these cysteine peptidases. You can't have cysteine peptidases just working away. They have to be activated. There's zymogens. Now, some can autocatalytic, depending on whether they're binding something, they can chew themselves and become active, when then they go down and start activating other ones. Now, if you don't believe in apoptosis or programmed cell death, a picture is worth 1,000 words. To me, this is absolutely remarkable. Embryonic development. This is programmed cell death in an embryonic mouse paw. This is day 12.5, day 13.5, day 14.5. Look at the restructuring that's going on. Next spring, when you see tadpoles, think about what's going to happen. In fact, I used to have some frogs, and I would bring them to class when they were just tadpoles, and people would come look at them. It's just stunning. It's stunning to see this happen. So here you go, a summary of what I've been saying. So in the case of the mitochondria, in the case of an intrinsic pathway, there's something that says the cell is under stress. It lets go of cytochrome C, and it forms this complex right here. This is called the apoptosis protease activating factor here. This comes and then interacts with caspase 9, which then self-cleaves, or autocatalytic, and then it starts coming down and it cleaves these procaspases, these zymogens, to give you active caspases, and then basically your proteins start getting degraded, and there's an orderly way of cell death. In the extrinsic pathway, there's going to be some death ligand or something that comes along, binds to these receptors. There are adapter molecules that recruit these caspases. They get activated, and then they come along and activate the caspases 3 and 7. No, you don't have to memorize it. What you do have to know is that programmed cell death depends upon caspases. There's compelling evidence that the mechanism of apoptosis is evolutionarily conserved. And just remember, we're still learning a lot about this. It wasn't that long ago when people didn't believe there was programmed cell death. But we now know. Now you can imagine, if you have too much programmed cell deaths, that something goes haywire, you can understand how you can start having disease states. Where if something, you're not getting cell death, cancer. Maybe you're not getting the cell death you need, so you get these cells just keep growing and keep growing. Most have a cysteine in their active site. Most cleave target proteins at a specific aspartic acid in that P1 position. At least 14 different mammalian caspases have been identified since 1993, which isn't that long ago. It isn't that long ago. All exist, as I said, as precursors. You can't have them chewing up unless it's time. And so some interact with adapters, and then they do this autocatalytic process where they cleave themselves. And then they go along and start cleaving other caspases along the way. So the caspases, they're generally divided into two groups. There's the initiator caspases, the ones at the beginning of that pathway, and then the effector caspases. So these are the initiator caspases, and these are the executioner caspases, or the effector caspases. And what you see with the initiator caspases is they have a bunch of other stuff at the end terminus. So this, the large and small subunit, that's the part that's going to be important for catalysis. But these, at the initiator, have some extra stuff that allow them to interact with adapter molecules, that allow them to interact and then become active by autocatalytic cleavage. And then they come along and start cleaving these. So what you see with the executioner caspases is they tend to be smaller. Activation of a caspase. So here you have the main chain, polypeptide chain here. There are two cleavage sites, and one's here and one's here. And what happens is you cleave all of this. This is the prodomain. And then what happens, you get this inactive precursor that can then get clipped to the large subunit and the small subunit, and now you have an activated caspase. It's pretty amazing. And if you're interested in how this activation goes on, there's actually a figure in this chapter, chapter 10, that explains how they think it happens right now, today. But who knows what will happen in a year from now. But the important point is, you start off with this precursor here, which is inactive. There's a cleavage right there. And then this inactive procaspase precursor gets clipped off to a large subunit and a small subunit. So there's two cleavage sites. And the preferred specificity of cleavage is a some residue, a glutamate some residue, and the aspartate. Caspases, aspartate. Now, for the longest time, people thought it had to be an aspartate in the P1 position, but we now know you can have a glutamate in that position as well. So I'm going to tell you about the human caspase 8. Now, why did I pick this? Part of it, it's medically relevant. There were people working on it that were involved in drug companies, but also I wanted to show you something about a reaction mechanism that they proposed, and this is where crystallographers aren't thinking in terms of their basic chemistry. So you may wonder why the first lecture I started talking about sp2 and sp3 and reminding you about basic chemistries, because there's a lot of people who have forgotten their basic chemistry. So this human caspase 8 activates downstream caspases, which are unable to carry out autocatalytic processing. So it can. So it's an initiator caspase. Its catalytic domain is 30,000 molecular weight. It comprises two polypeptide chains. Two. But actually, it has to come to two polypeptides to come together to actually form a tetramer, and I'll show you that in a minute. The alpha subunit contains the active site cysteine, and the beta subunit contributes to the formation of the active site. And its natural substrate is caspase 3. And its N-terminal prodomain contains these two death effector domains that bind to these adapter molecules and let it to become activated. So the crystallography of the human caspase 8 was solved, and it was autocatalytically processed. So they didn't have a prodomain and all the death receptors and everything. They just had it autocatalytically processed itself. And the protein was modified with this peptidic inhibitor, which is an isoleucine here, a glutamate, a threonine, and a aspartate. And look over here. Now, this is an aldehyde here. And what happens is with these kinds of compounds, and you'll see this in drug design, a lot of aldehydes are often used as inhibitors because when the cysteine attacks that carbonyl carbon here, and don't forget they were taking into account the specificity of the P1 site, the P2 site, the P3 site, the P4 site. Right? So when this attacks the carbonyl carbon here, you get a tetrahedral intermediate. But will that collapse? And the answer is no, because the cysteine, the sulfur is going to be here. That hydrogen, it's not going anywhere. It's not a good leaving group, and you can't protonate it to get it to go. And so you'll see these aldehydes used a lot of times to inhibit enzymes. So this is the structure of the alpha-beta heterodimer. So it's two subunits. There's the alpha subunit, which has the histidine and the cysteine right there, the thiolate, right, and the histine couple. And then there's the C-terminal domain, which is shown in red here. But we know that in order for it to be functional, it has to be an alpha-2, beta-2 heterotetramer. It's a good buried surface area. If you look at about 2100 angstroms, we've talked about buried surface areas, and that's pretty good. That's pretty good. So this is the structure that's important for catalytic activity. So they went ahead and solved the structure, and it is with this inhibitor bound in here. So you have the cysteine that's been bound here. This is actually tetrahedral here. There would be a hydrogen back here. And you can see that it's bound here and it's inhibited. Now, when you read these papers about peptidases, you always see these types of figures right here. And so I always say that if you know you're having trouble sleeping in the middle of the night, especially this time of year, and especially with the pandemic and everything, you just get up and look at one of these and you'll go back to sleep, aka cure for insomnia. Now you may find this boring, but if you're a medicinal chemist, this is not boring at all, because you're interested in understanding the interactions that can be used. So when you design something to inhibit it, you go, well, wait a minute, I know that there's an interaction with this aspartate and that arginine right there. And that's one of the reasons why you have an aspartate in that position, why it likes it. And what's interesting here is there's a histidine here, and then the carbonyl oxygen right here of the arginine. That helps in this, so the cysteine attacks here, but it's thought that that's important in activating that cysteine as well. So there's all these different types of interactions here. And this is what people that are involved in drug design want to understand. Now, they proposed a mechanism for this caspase, and I put it on this slide. And the reason why I put it on this slide, and the reason why I talk about this one, is because when you read the literature, make sure that you look at it carefully and realize that some people, they're not thinking correctly, that they're not thinking clearly. So this is their mechanism that they proposed, and chemically it makes no sense. So what happens here is our substrate coming in here. This is the carbonyl carbon that is going to get attacked. This is the Michaelis complex. Now they have a cysteine here, that's a thiolate, it's been ionized, and a histidine sitting here, that's protonated. So they have the cysteine attacking the carbonyl carbon right here, and you have a tetrahedral intermediate. Well, that makes sense, doesn't it? That makes a lot of sense. And they're saying that the protonated histidine up here helps to stabilize that oxyanion. Well, that makes sense. But wouldn't it make sense for the histidine to move the proton over to here to break this bond so you get a good leaving group? But instead, they invoked a water molecule just sitting there. And somehow this water molecule is magically going to take one of its protons and put it right there so you can break that bond and have a leaving group, and out comes a hydroxide. What are the chances of that are? They're not remembering their basic organic chemistry or their basic freshman chemistry. Now they have this acyl intermediate sitting here. Right? So now the histidine's protonated here, and somehow, magically, a hydroxide comes in here and breaks this off right here. A hydroxide. What? If you think about it, what makes more sense is that the histidine protonated is going to put a proton on right here. That's going to break apart. Now, a water molecule comes in. That does make sense. A water molecule would come in here, and then you could end up, this histidine is no longer protonated, a water molecule could come in here, and that histidine could activate the water molecule to get it to hydrolyze that acyl enzyme intermediate. But all of a sudden, magically, a water molecule is going to donate a proton, and a hydroxide is just going to come in. Makes no chemical sense. So the point of this is when you read papers, take a look at the figures. Be skeptical. Be curious. Be curious about what they're saying. So we'll finish off with the metallopeptidases, and as I said, the last lecture we'll talk about the aspartopeptidases, but the metallopeptidases, and they're very important. They're important in terms of cell proliferation, remodeling. They typically bond a zinc, but they can use other metals as well. Why is zinc used so much? Zinc is used for a structural metal too. Sometimes you solve a structured protein and the zinc is way off just stabilizing the loop. But zinc tends to be in active sites because it's not redox reactive, so you don't have to worry about oxidation and reduction. And zinc can have all different types of coordination spheres. So it can be tetrahedral, it can be trigonal bipyramidal, it can be octahedral. And so zinc you tend to see in active sites. Some of the most widely studied peptidases are the digestive peptidases that you know, carboxypeptidases A and B, thermolysin. Thermolysin, the one that I worked on as a postdoc with Brian Matthews. They are the most diverse of the 4 classes of peptidases, so the structures are quite different. What do? Metal ion catalysis. They can activate a water molecule for hydrolysis. So unlike the serine and the cysteine peptidases, where you end up getting an acyl enzyme intermediate or a thioacyl enzyme intermediate, these basically activate a water molecule and it directly attacks that carbonyl carbon, and you get the break of the bond. So it's more direct, and that's true of the aspartopeptidases too. They also activate a water molecule. And how do they activate the water molecule? When you have a water coordinated to a zinc there, the chances are it can dump off its proton, especially if it's in between 2 zincs, binuclear metal centers. If you have a hydroxide, you know that's a powerful nucleophile that's going to attack here. We also know that the zincs tend to, sometimes the substrate will come in and the carbonyl oxygen of the cis-alpha peptide bond will coordinate to the zinc, and that helps to polarize that carbonyl carbon bond right here, and that makes that carbon more susceptible to nucleophilic attack. So let's talk about the aminopeptidases. In the beginning I told you what an aminopeptidase is. They are exopeptidases, and they are removing the N-terminal amino acids from a protein. Some have 2 metals, sometimes they have 1, and usually it's zinc, because zinc, as I said, is not redox reactive, and it can adopt all these different types of coordination spheres. And as you might expect, when they go awry, they've been implicated in such conditions as cancer and HIV infection. And there's anti-tumor drugs, for example, ovolysin and fumagillin, that specifically inhibit these aminopeptidases. These 2 compounds are natural products. They are made by soil-dwelling bacteria. And the natural inhibitor B-statin, again, another one that's made out in nature, has been shown to significantly decrease HIV infections. So what do these structures look like of these inhibitors? And this is important to look at, because if you look at this one, B-statin, it kind of looks like a peptide, looks like the substrate, and C-alpha, C double bond O. But you can see that there's some differences here, right? So now we have a hydroxyl group sitting there instead of a carbonyl group. Now, that's B-statin, but fumagillin and ovolysin, if you look at those, well, they don't look like peptides at all. But they do inhibit these aminopeptidases, and they have these epoxy groups here that are very reactive. So we'll talk a little bit about this aminopeptidase from Streptomyces. As I told you, Streptomyces are gram-positive bacteria. They're soil-dwelling. Again, when it hasn't rained for a while, and then it rains, it smells like soil, it's Streptomyces. And these organisms make such an array of natural products. And there's so many out there we have not discovered. That's what's so exciting. So, they make, for example, the antibiotic streptomycin and the anti-cancer drug streptosin. The enzyme is calcium activated. It hydrolyzes peptide bonds formed by the N-terminal hydrophobic acids, such as a leucine, methionine, or phenylalanine. It's monomeric, pretty small protein. It has high enzymatic activity and high thermal stability, and it has a binuclear metal center that's required for activity. So here's the structure of this aminopeptidase. Nice structure, got some beta strands here, right? But here's the two metals sitting here, a binuclear metal center. Now the structure of this enzyme was solved, and the precipitant was phosphate. So if you look at the active site, this is a close-up view of the active site, and I'm going to take my time to just talk about this. It's a binuclear metal center. So here's one of the metals, here's the other metal, and this phosphate is sitting there. And normally what's thought is there would be a water molecule bridging there instead of a phosphate. But it was crystallized in high phosphate, so you have phosphates in there. But the phosphate serves to bridge these two zincs here. Now this zinc is coordinated by aspartate, 160, aspartate 97, a histidine, and then this bridging phosphate group. Now, this aspartate 97 is also acting as a bridging ligand. And in this case, it bridges this metal ion and this metal ion. And notice that the zinc is tetrahedrally coordinated. There's this tyrosine sitting there that's not interacting with the zinc. But we're going to talk about that. There's this histidine here, a glutamate here, and then this phosphate group here. Both are tetrahedrally coordinated. Now there's a glutamate 131 sitting down there and a tyrosine 246. So what's the proposed catalytic mechanism? Now, complicated slide, and if you want to see a better figure, look at this chapter, because the media specialist Laura drew it, and it looks a whole lot better than what I drew. But let's work our way through it. So here we have an example. Right? So we have a zinc sitting here, the two zincs, the binuclear metal center, and we have a water molecule. Now, is it a water molecule or hydroxide? And there's always people arguing it's probably hydroxide because it's stuck next to two zincs. Some people say, no, it's a water molecule. What do I think? I think it's probably hydroxide. But what happens is you start off with the zincs, and in comes the substrate. So here's that glutamate 131 sitting here, and this tyrosine 246. This is our zincs. The water molecule is coordinated to this. Now what the proposal has been, and notice when this binds, when this part binds, this is a cis-al peptide bond, when it binds, that carbonyl oxygen coordinates to the zinc, so it changes its coordination number. And that's the beauty of zinc, you can do that. And then there's this tyrosine sitting here, that tyrosine 246. This is going to polarize that carbon-oxygen bond here. Now what's thought to happen is, this glutamate 131 is going to help to pull that proton off the water molecule, if it is a water molecule. This is then going to attack the carbonyl carbon right here, and you're going to end up with a tetrahedral intermediate. So now the glutamate is protonated here. This hydroxyl group is going to go and attack the carbonyl carbon right here. You end up with a tetrahedral intermediate right here. This is called a gem dilate. So a tetrahedral intermediate, and this anion, the oxyanion, is stabilized by its interaction not only with that zinc, but also with that tyrosine 246. Now how are you going to break that peptide bond? You got to protonate it, otherwise it's not going to be a leaving group. And so the glutamate that pulled the proton off that water molecule is now going to deposit it onto the nitrogen here. And what's going to happen? You're going to end up breaking that bond, and out comes the peptide right here. You end up then, what's happening here gets protonated, that breaks, and you end up breaking the bond. So it's a very direct, we're going to activate that water molecule, it's going to attack the carbonyl carbon, you're going to go through a tetrahedral intermediate, and then that bond breaks, and out you go. You've done hydrolysis. So, a summary of the peptidases. They are biochemically and medically relevant. They are often targets for drug design. We still don't understand them all. Not one little bit yet. There's a lot to learn. But much of the enzyme chemistry involves chemical transformations of carbonyl groups in substrates and products. And that's true of so many metabolic pathways. If you understand carbonyl-chemistry, you understand biochemistry a lot better than what you think. There are 4 well-characterized mechanistic sets: the serine peptidases, the thiol or cysteine peptidases, the metallopeptidases, and as I said, the last lecture, we'll talk about the carboxyl or acid peptidases, and they work at acidic pH values. And finally, what to focus on. Know the difference between serine and cysteine peptidases. Know the difference. One is what is a catalytic triad, the other has a dyad. Know how the metallopeptidases functions, how are they inhibited, and what do the serine, cysteine, and metallopeptidases have in common? They're all going through tetrahedral intermediates. And you need to understand the Schechter-Bergen nomenclature, because we're going to talk about this again on the last day of class. So thank you, and as I said, the last 2 lectures are optional. I hope you come and listen to them, but it really, I know it's the end of the season, it's been a long haul. So please come. Thank you.